This lecture is going to be on the human eye. It's the result of the Cambrian explosion. You'll want to watch this video. This is really cool. It kind of depicts as to what took place as the eye evolved from single-celled organisms. Single-celled organisms has a light spot. Light-sensitive cells derived as early as primitive cells were evolving into complex structures. Being able to sense light gave an advantage to microorganisms who could find food or hide in the shade, right? Hide from predation. The eye was here to stay and evolve. So here's a primitive euglena and has a light spot, and it simply just picked up different amounts of light. So as the euglena would interpret light, it could actually travel towards it or away from it, which our eyeball is very much a direct evolutionary kind of difference, a change over time from this very light spot. And so our eyeball itself is detecting light. It actually makes a lot more sense of it. And here's how. The concave shape provides directional sensitivity. Directional sensitivity gives purpose to motion. An organism can be much more efficient when hunting or hiding if it can identify light signatures. So the concave shape here that we're looking at was the kind of the evolutionary feature of how the eyeball began to change. It actually gives a sense of where the light was coming from. So directional sensitivity here. And then a pinhole developed. The pinhole in the eye allows for finer directional sensitivity and limited imaging. And what happens here, the pinhole itself increased the resolution. It reduced distortion and gained directional sensitivity even more. So it actually allowed for, very specifically, as this little pinhole would allow light in, it actually, let's just say it made everything a little bit clearer. It allowed you to kind of take in tiny amounts of light, but make really good sense of them. And these are where our photoreceptors found on our retina would be here, located here in yellow. And then the next phase, we can talk about the iris fills with a fluid, and crystals created a lens. So the iris separates and comes to develop, and then your lens developed here. So in this case here, we have the parts of the eye that are beginning to evolve. Here we have the introductory part of the eye that allows light in. We have the lens that's going to refract the light, and we have the retina, which is going to then make sense of the light in both intensity and in color. So the lens refracts light onto the retina very efficiently. The eyeball has a great adaptability of near and far vision. And here you can watch a video on concave and convex lenses. I'll show you real quickly here that you'll see that the different types of lenses actually allow for light to be refracted different ways. So in this case here, well, I'll just tell it myself.
Concave and convex lenses. Notice that a lens allows for light or photons to pass straight through. But if I move towards a concave lens, notice the greater the amount of concaveness is, the greater that light refracts. And notice that it bends outward. It bends outward because it's refracting as it moves through the concave lens. That if I have a convex lens, it's going to be converging. So notice the convergence. I'm changing the location of the focus. So here I have the changing of my focus with convex. Once again, they converge. Convex converges. And concave diverges. Diverges means moves away. So I have divergence here. The more concave it is, the more divergent. And the more convex, the greater the convex lens, the closer the foci is to the lens itself.
So in summary, remember, lenses allow light to pass through them, and so therefore as light passes through the new media, it's going to refract. A converging lens bends rays toward the focal point, right? In this case, puts all the light in one location, and a diverging lens bends rays outward or away. It spreads them out. So since we're talking about the eyeball, we're talking about, in this case, a converging lens. In this case, we're talking about a convex lens, right? Convex converges. So when I look here, we can really apply the rules of refraction. We can see that the light traveling from air into, in this case, we can say glass, it's going to refract the light, and everything's going to be measured relative to the normal line. If it goes from low to high, it's going to refract towards the normal line, and when it goes from high to low, it's going to refract away from the normal line. And in both cases, the curvature of the lens is going to dictate where the focal point, where it comes back together. And so make sure you read through here 1 through 5, but the net effect shows how light refracts into one focal point onto the human retina, where we make sense of that light. And we can make sense of this, therefore, in theory, we want that focal point to be right on our retina. And the retina then has photoreceptors, which I'll get into in a minute, cones and rods, and that's what makes sense of the light. But not all eyeballs have this perfect lens that refract the light to the retina. Some people are farsighted. So therefore we need corrective lenses. For farsightedness, one would say that the eyeball is actually too short, that the focus is behind the retina itself. So you would use a converging lens that is then used to correct the condition. The lens refracts the light entering the eye to focus onto the retina. And that's what contacts or glasses would do. It changes the location of this focal point to place it directly onto the retina. And whereas somebody who is farsighted, the eyeball is too long, that you would actually need a diverging or a concave lens that could be used to correct this condition. The lens refracts away from the principal axis before entering the eye, and then the rays are able to focus on the retina. So you want to spread this back a little bit. And so remember, if we're going to change the focal point here, if we want it closer, we're using this type of lens, a convex lens. And here we'd use a concave lens for farsightedness. And so at the end of the day, the human eye evolved. It started off as these simple cells, and we came a long way. Notice, region of photosensitive cells. It then changed and folded and opened up a region for directional sensitivity. The pinhole allowed for a greater ability to make sense of the light. We have this fluid filled, right? The transparent, it's called the humor, develops in an enclosed chamber. We have the retina developing and evolving. We have a lens that develops. The lens is going to then refract the light onto the retina itself, where it can further make sense of what's going on. And this information is then sent to the optic nerve to the brain. So therefore, the detailed infrastructure of the eyeball, it's pretty complex in there. There's a whole lot going on. But here's the key to this. As our eyes got better, our brain got better. A brain that can process light information changed the visual cortex and our ability to see more and better. So the eye evolving actually started off as a primitive cell from this euglena, right? Just from this light spot, has evolved into us being able to make sense of light in a lot of different ways. And we make sense of our world, generally, for the most part, through our ability to see it. So where does light come from? Well, light is either emitted, given off as a result of an energy transfer, or is reflected off a surface. Here are two different examples that we're most familiar to, right? Light bulbs and the sun. They give off light. But light moves in straight lines as rays and bundles of energy called photons, right? These straight lines. Light moves in straight lines. Each photon has a specific quanta that it must abide by. And we see our world mainly by reflected light. When you're reading a book or looking at somebody's shirt or whatever it is you're viewing, generally you're seeing light that strikes an object and reflects off it and makes its way to your eye. So then that's where you create that image as it goes through your eyeball itself. Well, let's look at what light is. We have to look at the electromagnetic spectrum. Light travels at c in the vacuum of space. C is 186,282 miles per second. That's pretty fast. The E&M spectrum illustrates the different forms of pure energy. Observe, radio waves have the largest wavelength and the lowest frequency. Gamma rays have the shortest wavelength and the highest frequency. And when we go from long waves to short waves, we would say radio waves, then microwaves, then infrared, then visible light. Remember, Roy G. Biv, these are the colors of visible light. We have ultraviolet, X-ray, and gamma. That, when we look to the right, the visible light is simply just a piece or a part of the electromagnetic spectrum. White light can be separated by a prism into all of the colors. And so visible light follows the same rules, right? Red has a large wavelength that would be on this side of, if I looked at it, the electromagnetic spectrum as a total and included the visible light, and therefore blue and violet would be on the shorter wavelength side. So when I look here, we're talking about the wavelength and the frequencies. The frequencies here, the smaller the frequency, the smaller or the lower the energy, and proportionally larger the wavelength. And therefore your higher energy side would have shorter wavelengths and higher frequencies. So what are photons, right? Really, what is light? Well, white light is comprised of all of the colors of the rainbow. A rainbow is where water droplets refract the light, acting as little tiny prisms and separate the white light into the rainbow that we see. But it's really separating the photons. The photons are the bundles of energy. Colors of light are pure energy and cannot be broken down any further. Colors of light vary in their frequencies and wavelengths. And all colors of light and everything on the E&M spectrum travels at c, the speed of light. That if I only compare red photons and blue photons, really the only thing that differs would be their frequencies and wavelengths. Red would have a larger wavelength and a smaller frequency. Blue would have a higher frequency, right, larger frequency, and a smaller wavelength. But they are both traveling at the speed of light. And here we can compare the Roy G. Biv on terms of a frequency and wavelength scale. And you can actually see that red has a larger wavelength than all other colors and a smaller frequency, and whereas violet has a smaller wavelength and a higher frequency. So blue and violet are the highest energy, red and orange are the lowest energy, and these are generally based on the frequencies. So what takes place within the human eye? Well, process of seeing color, or pretty much your world, it starts with the pupil. The pupil is what dilates and contracts based on available light. This is what lets light in. Then it refracts through the lens, right? The lens is a part of the opening of the eye, refracts it onto your retina. The retina is comprised of cones and rods. Cones allow you to indicate color. The brain, or the eye itself, the photoreceptors in our eyes are red, green, and blue. The rods indicate or pick up intensity, light intensity, how much light is coming in. Then this signals a message from the optic nerve. The optic nerve is then stimulated and sends a message to the brain. And all of this happens literally in less than a snap of a finger. It happens very quickly. Our brains and our eyeballs work as these supercomputers that process tremendous amounts of energy. So we see reflected or emitted light. However, it's not as simple as that. We have to look at what we call the additive and the subtractive primary color, so the color processes. The additive color process involves mixing colors of light. Primary colors are red, green, and blue. Secondary colors are cyan, yellow, and magenta. Adding primary with a secondary secondary color leads to white light. We would say that they were complements of one another. So mixing red and green light creates yellow. Mixing yellow and blue, one gets white light. Remember, yellow and blue, those are complements. They create white. The additive color process involves light, where the subtractive color process involves reflected light off of a substance that contains pigments. Pigments absorb some of the light and reflect different light. In other words, it subtracts out some of the color as it absorbs it. For example, when white light shines on a red shirt, cyan is absorbed, right? Blue and green from the white light. And they are subtracted from the white light. Reflecting, the only color reflecting is red. So when white light shines on a shirt, it's going to absorb some based on the pigments. It's going to only reflect the color of the shirt itself. So we see the color because that's what gets reflected off of it. So let's put this all together. Additive and subtractive color process. The reason why we see green, say from a plant leaf, is because the colors red and blue are absorbed, right? When we know that red and blue together, it's simply absorbing magenta. And the color that gets reflected off of it is only green itself. So green is reflected. Here, we're seeing if we're mixing colors, we're involving the subtractive color process. So the subtractive color process, notice we have white light shining onto this color here. This should be cyan. Cyan is going to absorb or subtract red. White light, notice red, green, and blue all make up white light, shining on yellow ink or yellow pigment, yellow paint. Yellow absorbs or subtracts the blue from the light. And if we mix these two colors together, the mixing of cyan and yellow actually subtracts both red and blue. So only green from the light remains. So if we mix these two colors, we would actually see it as green. And that's an application of the subtractive color process.